Hello everyone, in this video I'm going to be talking through synaptic transmission. Now this is an A-level biology video. I have done a video previously on neurons and the synapse, but that one was for Key Stage 4 for GCSE. So this one's going to go into a little bit greater depth, particularly about what happens when the sodium and calcium channels open. And we'll go a bit more involved about the actual neurotransmitter also. So what we've got at the top of the image, over to the left here, and I've left space on the right for notes. What we've got on the left is one neuron essentially connecting to another, one nerve cell connecting to another one. And this is an excitatory neuron that we're going to be referring to, because there's different types. There's inhibitory, excitatory, there's a synapse to a neuromuscular junction, or to muscle essentially. So if we think of a basic neuron, nerve cell, we've got an axon, and then at the end of the axon, what we would say is we've got the axon terminal. Now the axon terminal finishes with the synaptic knob, a sort of enlargement or bulbous part of the axon terminal. As well as synaptic knob, it's sometimes referred to as the synaptic bouton, B-O-U-T-T-O-N. So I might as well just put, where I've got axon, I might put in brackets here terminal, because this is the end of our axon. And you can see we finish with this enlargement, this synaptic knob. Then we have a gap. Now that gap is known as the synapse, or sometimes referred to as the synaptic cleft. And then you can see it's making a connection with a dendrite. That's the top of another neuron. So an axon terminal makes a connection with the dendrite. Now, because these are neurons, nerve cells, we can see that they've both got cell membranes. Now these are important structures in this process. This one here we're going to call the pre-SM, synaptic membrane, because it comes before the synapse. And we're going to call this one here the post-SM, post-synaptic membrane. So we've got the two membranes there. We've labelled the dendrite, the axon, and we've got labelled as well the myelin sheath around the axon at the top, just for completeness sake. But there are two other really key structures that I need to mention. So here at the top we have, and I've not drawn them all clearly, we've got a mitochondrion. Now mitochondrion release ATP in aerobic respiration, and we're going to use that ATP to drive some of the processes. And what we have here in sacs, and again I've not drawn them all, these are membranous sacs called vesicles. And these vesicles contain what's called neurotransmitter. Now the example of the neurotransmitter we're going to talk about is acetylcholine, sometimes shortened to ACH. So this nerve transmitter, this neurotransmitter, is within these vesicles. I'm drawing that as yellow dots. So the first thing that happens, if we were to write this as a sequence of events, the first thing that we've got happening, number one over here, is that the impulse reaches the axon terminal, or towards the essentially synaptic knob. So we've got an impulse, and this is an electrical impulse, reaches the synaptic knob. That's our first stage. And what we get is essentially this action potential forming. Now, I have done a video that goes into much more detail about action potentials within neurons. I look at action potentials in terms of depolarization, repolarization. I'm not going to really deal with that in this video. I'm purely just talking about essentially what happens at the synapse. But for more information on action potentials, there is another video for that. So this action potential arrives at this synaptic knob, and what that does is cause calcium channels to open in that synaptic knob. So if we just draw these as two kind of yellow dots here, or yellow parts, what we get after we've had the impulse travel down this axon terminal, reach the synaptic knob, it causes this action potential wave of depolarization, and these calcium channels open. And calcium ions, Ca2 plus calcium ions, pass through that channel. So Ca2 plus ions pass into the axon terminal, through those calcium channels. So we'll just have that then. So Ca2 plus channels open. Now when calcium rushes in, what that does is cause these vesicles containing neurotransmitter to move down to the base of the synaptic knob and fuse with the presynaptic membrane. So these vesicles move down and they fuse with this presynaptic membrane. So I'll put that in place there. So the vesicles fuse with the presynaptic membrane. Now once they fuse, they actually burst, and that neurotransmitter, that chemical within them, gets released down the synapse and across the gap, and it literally just drops across the gap. So these yellow dots I'll just draw here. So after the vesicles are fused, these yellow dots essentially just drop down that synapse. So let's make a little note there. I'm just use different colours just to mix it up a little bit. So the neurotransmitter, and I'll put there EG because the one we're going to talk about is ACH, which is acetylcholine. What that does, it gets released and diffuses across the synapse. So let's put that one in place there. Now, when you look at the postsynaptic membrane, what you find is that there are actually receptors on this postsynaptic membrane, receptors for this neurotransmitter, which, again, not to scale, but we're just going to colour this in blue here. So these are special receptors for that neurotransmitter. Now, when that neurotransmitter chemical binds to those receptors, what it does is cause the opening of sodium channels in the postsynaptic neuron. So the moment that neurotransmitter binds to the receptors, sodium channels, I'll just put this here in blue, sodium channels open and sodium ions rush in. And when sodium ions rush in, that causes depolarization within that particular neuron, and we get an action potential set up, and that essentially allows an impulse to be generated in the second nerve cell, which can then be transmitted down that cell. So neurotransmitter binds to receptor, sodium channels open, sodium ions rush in, action potential set up, impulse can travel down that postsynaptic neuron. So let's put that particular note over to the right hand side. So after it diffuses across the gap, if I just put NT for neurotransmitter there, it binds to the receptor that triggers the entry of Na+, that's sodium ions, Na+ ions, and that essentially causes an action potential to be set up. Now what we need to think, had to finish this essential process, is to consider how we're able to go again. What happens really once this happens? Because we've had vesicles bursting, all this neurotransmitter essentially needs to be reformed. So that's exactly what happens. There is an enzyme within the postsynaptic membrane that breaks down this neurotransmitter. Now in this example, the enzyme is called acetylcholine esterase. Ends in ASE, esterase, so it's an enzyme. So you've got this acetylcholine esterase enzyme that will break down this neurotransmitter at the receptor. And then we use the ATP released from the mitochondrion to actively pump that broken down product, the broken down product from the neurotransmitter, back into the synaptic knob. Once that's been actively transported back into the synaptic knob, it then gets repackaged into another vesicle. So we're almost doing this in reverse. We're kind of getting ready to start this process again. So acetylcholinesterase has broken down the neurotransmitter. It goes back into the synaptic knob, gets actively pumped back in. It gets repackaged into a vesicle. Then all we've got left is to deal with the calcium. And then, once we've been able to repackage the neurotransmitter into the vesicle, the calcium ions get pumped out of the synaptic knob. Once they're pumped out of the synaptic knob, the process is then allowed to go again. Another impulse will essentially work its way down this presynaptic neuron. That will trigger calcium channels to open, calcium ions will rush back in, vesicles that are newly formed will then go down and fuse with the presynaptic membrane, burst, release the neurotransmitter down the synaptic cleft, and so on and so on. So I'm not going to essentially write that bit after number 4 just to save space, but once we've essentially had the action potential set up in the postsynaptic membrane, or rather the postsynaptic neuron, just to reiterate, what we have is the neurotransmitter chemical broken down by an enzyme on the postsynaptic membrane. In this case, it's acetylcholinesterase. The broken down product gets actively transported back into the synaptic knob where it's repackaged in a vesicle, and calcium ions, Ca2+ ions, get actively pumped out of the synaptic knob so that the process can then start again. And this whole thing that I've described here is essentially synaptic transmission across the synapse, this gap or junction between two neurons. Okay, hope all that helps.